Thank you much, Bobby. And I will reiterate Sheena's thanks to Anne and Greg for organizing this, for inviting me to be a speaker. It's an amazing time to be able to participate in research at the Lemur Center, and especially to be another odd man out in the research world. 2 weeks ago, I was at a conference for gut microbes, and I was the only researcher studying lemurs because, you know, that's me. And today, I get to be in a room surrounded by other people who love and appreciate and study lemurs, and I'm the only gut microbial person speaking. So it's a nice little flip of the coin. So it's pretty exciting. So I guess I'll kind of out myself starting there, that we've been learning a lot today so far about all these different phenotypes and behaviors of lemurs. And when I see a lemur, I see a bag of bugs. So with that, I would say I refer to myself as a microbial primatologist. This is kind of a term that I coined for myself this spring. This is my logo, now called the SCAT signal, because I collect a lot of feces to study gut microbes. And just with the idea that microbes make us who we are. We as animals evolved in a world that has always been bacterial in nature, way before multicellularity evolved, so you can imagine that these bacteria are involved in every aspect of our lives. Who we are as individuals, who we become as species. So just to kind of throw that out there at all of these different organizational levels, at all of these different time spans. And life naturally then depends on microbes. If you evolve in this environment that's filled with single-celled organisms, and those single-celled organisms become integrated into every life process. So along an individual's lifespan, this might include your brain development, your gut development, even in utero, and then your health once you have been expelled from that uterus into the harsh environment outside. Who you are as an individual and your health is dictated and tied up with the microbes on, in, around you, and especially the gut microbes naturally. That's why I'm here. Your gut is your primary interface with the world. Everything that you take inside, that you put in your mouth, so think back to your snacks, your lunch, your breakfast, everything. That's a sample of the world around you that you're putting inside your body. So your gut becomes not your skin, not your eyes, not your ears. Your gut is your primary interface with the world around you. I like to think of the gut as the second brain. So you have these 100 trillion microbes living mostly in your lower gut, right? You too. Even you, not just these lemurs, and they are the ones that are helping you to navigate this world that you're putting in your body in the form of meals. So, in addition to helping to break down all of the nutrients that you put into your body, especially fiber, which we'll get to in a minute, the products of digestion that those microbes are producing inside you become chemical signals that affect the moods that you have, the foods that you crave. Your gut may be your second brain, but the bacteria are really the drivers behind a lot of what makes you you. So if that doesn't convince you, then I don't know. So just to kind of map this out, these bacteria in your gut are affecting you, but there are certain host characteristics that are also helping to shape these complex bacterial communities, specifically the host gut and the diet. The gut becomes kind of the topology, right? That entire ecosystem, all of that ecological niche space for these microbes to have a place, to live, to eat, to thrive, or whether they're expelled, that's partially determined based on the gut complexity and the transit time, like how fast is that river of nutrients cruising through your body. And also the diet. Everything you eat is also feeding your microbes, especially the fiber, because you as a host can't digest that nutrient. So think about that for a second. For humans, it's not that essential. We're only getting 8 to 10% of our energy requirement per day from our gut microbes digesting that fiber. But things like cows and every other herbivore on the planet, they wouldn't exist without their gut microbes. So naturally, that becomes very crucial for some of these species of lemurs. So the host gut and the diet are both forming the gut microbial community, and I'm interested specifically in the aspects of the structure and the function of these communities. So I'm talking about structure, the community membership, as measured by sequencing a specific target or fingerprint gene. So instead of using these mitochondrial genes like we would for hosts, I'm looking at the 16S gene region. And then in order to get a handle on the functional profiles of these communities, I'm looking at the metagenomic functional profile, so that's shotgun sequencing. It's chopping up all of the DNA from a single fecal sample and then sequencing small bits that are coming from all of the organisms that make up that entire community. So I'm looking at structure and function at the community level and looking for relationships between these different bacterial aspects. And finally, looking at how those microbes are actually utilizing and digesting that diet through a process called fermentation to produce digestive components like short-chain fatty acids that comprise nutrients that the host, yourselves included, can actually absorb as an energy source, but also that become chemical signals to that host. So the system, back to the lemurs. This is your picture of lemurs in this talk. So I'm studying and comparing in this talk 3 different species of lemurs that are highly diverged and that actually exhibit a range of different host genetic related different morphologies and characteristics. So here I'm talking specifically about the gut morphology. So you have Varicea variegata that has this pretty short GI tract and a fast transit time. The ring-tailed lemurs have a more intermediate GI tract, so they would be kind of the closest complexity of the gut to a human, right? About 7 times the length of their body compared to ours, about 7 to 8 times the length of our body. Whereas the Propithecus cocorelli, these shafaks, they're kind of the cows of lemurland or the horses of lemurland. They're eating all leaves all the time, so they need a long GI tract that's about 16.6 times the length of their body. And the food is going to hang out in there a long time, which gives the gut microbes a lot of time to process all of that fiber. In captivity at the Duke Lemur Center, the Varicea and the Lemur cata are actually fed just about the same diet, a mixture of fruits and vegetables. So I'm referring to them as frugivorous because vegetables are still technically fruiting bodies. So bear with me there. Compared to folivorous diets, which are these high-fiber leaf-based diets. So I sent off samples of each of those diets, dried and ground, to the Cumberland Valley Analytical Services to demonstrate that these diets are providing different nutritional profiles to the gut microbes. So again, you see this kind of frugivorous diet has very high levels of starch compared to the folivorous diets fed to the shafaks, whereas they have lower levels of the neutral detergent fibers and the acid detergent fibers. So that's your hemicellulose, cellulose, and lignin. So those are your structural components that really give plants their toughness, right? That's that fiber, the structural component. So sampling these fecal samples from all of these different infants, I collected feces and had a lot of help at this stage, well, at many of these life stages, from Bobby, Kathy, and the keepers and staff at the Lemur Center. This slide should actually be tied directly to my acknowledgement slide because couldn't have done it without all of you monitoring the animals to find out exactly when these animals are born. Where else in the world can you get a baby lemur in hand for a tiny fecal swab the day it's born to figure out what's going on in its gut from birth all the way to adulthood to see how these communities are forming over time. So I collected and had a lot of help collecting fecal samples from each of these 6 life stages of development from infants belonging to each of those species, and then also compared to the mothers during the birth process, which we call parturition. And I had 2 big questions with this. One is, how does this microbial community change over time in an infant? So when you're going from just about nothing in the gut to over 100 trillion microbes in the gut, how does that community build? Does it follow a similar succession trajectory pattern within species, or do you see just vast differences between individuals? My second big question is, do these different lemur species host different communities? So step one is the identity check. Again, using the 16S gene target to identify which specific types of bacteria are making up these communities. And what you would get, we identified hundreds of different genus-level bacteria. So instead of showing you a bar chart, which would be just a huge mess, I'm providing you with summary statistics here, and then we'll dive into what makes these patterns. The two big takeaways here are that the diversity increases with age. So green is going to be your Propithecus, eating those leafy greens. The orange and red are the Varicea variegata and the Lemur cata, both eating that more frugivorous diet. The big takeaway here, you do have diversity increasing with age, but the Propithecus are always ending up on top, right? So the more fiber that you eat, it's seemingly culturing or cultivating a more diverse bacterial community in the gut. But despite that higher diversity, there is less inter-individual variation between those leaf-eating lemurs' gut communities. And this becomes even more apparent when you start to look at these data projected in a different way through a principal coordinate analysis. So each of the dots on this graph represents a different gut community sampled from a different individual lemur infant at a different life stage. The major takeaways are all the way up the y-axis on the far right. Those are your birth and nursing samples, huge amounts of variation early in life. Then that middle section, as soon as they start to eat solid foods, you see a tremendous shift, a convergence toward the climax communities that are identified in the animals that are adults, that are even just eating solid foods on a daily basis. So these are the core communities by each host species that you would associate with healthy adults. And the major takeaways there, again, you have the Varicea variegata and the Lemur cata that are eating the same captive diet, and you see a large overlap between their community structures, but still with a bit of variation that I think may have to do with their shorter GI length, faster transit time. That means less regulation power by the host to select for specific communities, compared to the shafak. Teeny tiny circle, it's highly conserved. You have a lot of reproducibility between these different individuals, that it's associated with this high fiber diet and a long GI tract, which I suspect gives the host plenty of time to regulate exactly who is in the gut. So if we see this type of regulation, then the next question becomes, what does that mean functionally about those different communities? Using linear discriminant analysis for these Propithecus cocorelli to figure out which specific bacterial types are setting those unique communities apart, Prevotella, Ruminococcus, and Oscillospira are all fiber digesters identified in the literature. So the next natural step then seems to be, if these folivores are recruiting diverse specialists, then what does their functional profile of these microbial communities look like? So step two is to check out those microbial skill sets associated with these guts. So again, it's a shotgun metagenomic approach, so a huge amount of data. Instead of just genes from one region in the genome from only the bacteria, this gives you a sampling of bacteria, fungi, yeasts, protozoans, protists, any viruses that are hanging out. You have to filter out the host data because I only want to look at the gut microbiome. And it's all the genes that are potentially associated with that entire community. So again, we'll simplify, starting out with a principal coordinate plot. And hopefully this is apparent. You see, regardless of all three species being included, a clear distinction between the folivorous and the omnivorous diet fed in captivity that seems to split these three species even across life stages. When I then looked for which enriched genes belong to which metabolic pathways that might underlie these patterns, we see that diet seems to select for differential path enrichment. So you can see that the leaf eaters tend to cultivate gut microbiomes that are enriched for amino acid production and metabolism, for nucleotide production and metabolism. The amino acids, I am in the process of looking into whether those are essential amino acids. I would assume they would be, that the microbiome might be very important for supplying those essential amino acids, especially to herbivorous animals. That's what we tend to see in other herbivorous species. The nucleotides, I think, have to do with microbes making more microbes. So if you have these highly diverse communities in a very long and stable gut community environment, then these microbes would need to maintain that high diversity over time. So microbes making more microbes. We know that gut microbes are important for developing different cofactors and vitamins that in turn supplement human and other host nutrition. But I really want to focus on these carbohydrates because I tend to think of fibers as being highly complex, and we saw that they are recruiting a very diverse number of different microbial organisms. So then why am I only seeing 26 enriched pathways compared to 36 in the omnivores, Varicia variegata and Lemur cata? So when I plotted those specific metabolic enzymes and metabolites on a KEGG metabolic map, what I'm seeing at the top, you have in pink, those are the enzymes and different genes that were enriched in the omnivorous lemurs, and those are associated with sugar and starch digestion. Whereas in green, for the folivores, you see those are the ones that tend to be involved, especially down here. This is associated with anaerobic fermentation, often of fibers. So we would expect in a very long GI tract for those microbes to have to function in an environment with little to no oxygen. So I'm actually really excited about the TCA cycle results there. Also, when you think about sugars and starches, they're very small and multitudinous, right? So you have very short, very different ways to be a simple molecule like a sugar or a starch. But fibers tend to all be very long with double bonded chemical structures and branches. So they tend to have conserved structures. So it starts to make sense that you might have conserved functions for breaking down those different structures. Also, this little cluster of green over on the left, that is a pathway for metabolizing phytic acid, which is a secondary plant compound, which plants actually produce to keep animals from eating them. So I'm pretty excited about that result as well because it's suggesting that these gut microbes may be very important for these lemurs to be able to digest such a leaf-rich diet. So skill sets are kind of like looking at someone's CV, but they're not actually telling you what job they hold currently. So in order to look at what job these microorganisms are performing in the guts of these different lemurs, I then sent off little shavings of these frozen fecal samples to the David H. Murdoch Research Institute in Kannapolis. I have a collaborator there who ran NMR analysis to quantify different metabolites, including the short-chain fatty acids, which are the products of microbial fermentation, that the gut microbes are actually producing from starches and fibers that are coming in with the diet. And what we see here is not only do we see some differences between Varicia variegata and Lemur cata, even though they were eating this a very similar captive diet, but also we see higher total short-chain fatty acid production in these leaf eaters, especially of acetate and butyrate, which are short-chain fatty acids that are produced specifically from those fiber substrates, whereas propionate tends to be a microbial product that's fermented from starch. So fermentation is here reflecting the dietary composition. So again, you have you can kind of connect these slopes across the graphs. You have less starch in these leafy greens being eaten by the shafaks, and in turn you have less propionate being produced by those gut microbes. Conversely, you have more fiber coming in with the diet, and you have more acetate and butyrate being produced by those lemurs' gut microbes. So, to come up with a single graphic to put this all together, it really seems as though these 4 disparate pieces of the puzzle all come together and they fit. So to give you another data visualization that's actually based on my results, that kind of brings to light this puzzle piece, fitting together, I give you microbial geometry. So this is the idea of relationship between these 4 different pieces of the puzzle. So we'll start with green for shafaks, as always. If you are a leaf eater, starting at the top, you are bringing in a higher % of dietary fiber. That is supporting higher bacterial diversity, which are able, on the left, to produce a higher profile of short-chain fatty acids. They're fermenting more products from all of that fiber coming in. However, as we saw in the KEGG metabolic pathway enrichment, they're actually showing functional redundancy. So you have more different types of bacteria creating more things using a more narrow range of the functional potential suite. So, and you have very different shapes for your leaf eaters compared to kind of your frugivores eating that fruit and vegetable mix in captivity. So, if you want to know more, now that we have kind of established the gut community and how it's established in health from birth to weaning, Lydia Greene and I have been collaborating to collect samples from shafak infants. Once they're weaned, they become highly susceptible to an infectious pathogen called Cryptosporidium. And the results that we find, you may have heard of personalized medicine. Well, she can tell you all about personalized disease and these individual trajectories that we're seeing in infected lemurs. So she has poster 23 tonight at the Lemur Center. Be there. It'll be awesome. And finally, my many, many thanks. The Duke Lemur Center and NSF both funded the work that I presented today. My committee members are amazingly positive. They're brilliant and just exceptionally supportive. The Duke Lemur Center staff, Erin McKenney, David Brewer, Bobby Shopler, Kathy Williams. Bobby, you're up here twice because you're doing double duty. Thank you so, so much for all of that help collecting, taking these lemurs in hand in health and disease. Lydia and Tom, my 2 collaborators, and to the labs that did all of the sequencing, and my support from the Yoder lab, past and present. So, thanks.